Hi, everyone, and welcome to today's lecture on DNA replication, repair, and recombination. We are at the part of biochem that is my favorite since you guys all know I love that DNA, which, come to think of it, sounds kind of inappropriate and dirty, but you guys know what I mean. I love anytime we get into the genetic stuff. So hopefully, you guys will be as interested in this stuff as I am. So the first stuff that we're going to look at is DNA replication. And first off, I want you to always remember that DNA replication is considered semi-conservative. So this figure shows that, when they were first thinking about how DNA replication is happening and what's going on, there were three options of what it could be. It could either be conservative replication, where you see you have the double-stranded parental strand, and when you make the daughter molecules, it's just a separate new double strand of DNA. So both of the new double strands are in the daughter DNA. There's dispersive replication, okay, where you see each daughter DNA molecule contains a mixture of old and new. And so it's a little tough to tell in this figure, but basically in this, each single strand is alternating. So it has some old, some new, some old, some new. That's why you see blue and green on a single strand. But what it turned out to be is semi-conservative, where you have the parental double strands separate, and so the new daughter molecules each contain one whole original parental strand plus one whole newly formed strand. Okay, so both of the daughter molecules have one strand old and one whole strand new. And they discovered that through Meselson and Stahl. So the whom is Meselson and Stahl, and this was their experiment over here, where they basically gave biochemical evidence that the DNA replication is semi-conservative. So they grew bacteria, specifically E. coli, in 15, so radioactively labeled 15 nitrogen medium. So that's basically the heavy DNA, the heavy labeled DNA. And then they switched the bacteria to light isotope DNA. So they call that light DNA. And what they found is when the nitrogen is the 14 labeled isotope and you have replication occurring, so the bacteria is gonna have to use that nitrogen to make the new strands. You had the semi-conservative look of, one old strand, one new strand together. And then in the final later generations, you ended up with the fully light because, the light strand separated from the heavy strand and each became a template for new DNA. So you had the fully light one made and the hybrid one made. And the only way that you could have a hybrid at this point is if you still had one strand of that previous parental DNA getting carried on to the new DNA. So when we look at the process of DNA replication, we're going to get into some more details in a minute. But the first thing I want to point out is that DNA replication requires what's called an origin of replication. And so over here you see it replication origin. And basically, that's a sequence where it's going to be a recognition site for the enzymes that are responsible for DNA replication. And so when you have that origin of replication and you have the enzymes coming in, the proteins coming in that actually do the process of DNA replication, you need then the formation of a replication fork where you have the two strands of DNA separating to open up and to give you room now to use each of those strands as a template to make the new strands of DNA. Okay. Now, when you look at the direction of this process that's occurring, at first they thought, oh, it could either be one direction or, sorry, laughing at the band, or bidirectional. And they determined ultimately that it is a bidirectional direction that it occurs. And so you end up having those two parental strands separating, each being a template for the new strands, and those new strands are getting formed concurrently, meaning at the same time in different directions. Okay? And both of them, ultimately, the direction will be 5 prime to 3 prime. Okay, so now I want to give you a little more detail into the process of bacterial DNA replication. And again, so notice that this is bacterial. Something that bacterial DNA replication requires that we do not is called gyrase. And so gyrase, you see that here, that's necessary in bacteria to uncoil the DNA because currently at that moment, the DNA is all coiled up into a tight little ball to make sure that it kind of fits inside of that cell. Now, what's very valuable to us in the medical industry is that bacteria have gyrase to coil and uncoil their DNA. We do not. And that's very valuable to know because that makes gyrase what we call a high or highly selective toxicity target for drugs and antibiotics. And what that means is gyrase has high selective toxicity because it's something that we can target to kill or hurt the bacteria without hurting the human cells. So that's what we mean by selective toxicity. You want to hurt the bacteria without hurting the human. And so gyrase is great for us to target. Now, once the gyrase has uncoiled that, big ball of DNA, the next thing that you end up needing is helicase and SSB. SSB stands for single strand binding protein. You'll see that written out in the chalkboard notes next. Now, the reason you need these two enzymes, first of all, the gyrase in the previous step had uncoiled that coiled up condensed ball of DNA. The helicase now unzips the double strands to expose the naked or separated little single strands. Now, if you did not have what's called SSB to quickly bind and coat those single strands, what would those single strands want to do? Well, anything that's single wants to find its mate, right? It's very clingy and wants to stick to its complementary pair. So those two strands that are complements of each other, once you unzip them with helicase, they would want to religate. They would want to stick right back to each other. And so you would not be able to have the rest of the enzymes come along. You wouldn't have DNA replication working without SSB to prevent the religation of those two strands. Now, once those strands are separated, the polymerases can bind. And DNA polymerase, when you hear the word polymerase, that means that it is going to build a new strand of nucleic acid. And if it's DNA polymerase, it's going to build a strand of DNA. If it's RNA polymerase, it's going to build a strand of RNA. Now, DNA polymerase has two limitations. First of all, it is one directional, meaning the DNA polymerase can only build in a 5 prime to 3 prime direction, meaning that its first base would have to be 5 prime and the last base would have to be in the 3 prime end. Okay? The second problem is that it cannot start new strands. Okay, it can't start a strand from scratch. And to compensate for those limitations, what you need is called primase. Primase is a special RNA polymerase that will make these little blue segments here. Those are RNA primers. And we said primase is RNA polymerase, meaning that it's going to put RNA bases at the beginning here. Once the primase has made that little primer, now the DNA polymerase has something to attach to. to attach the new bases to. It has a 3' end to start sticking DNA bases to, because we said it cannot start a strand. So it needs those RNA primers first. RNA polymerase is basically a rock star. It does not have the limitations that DNA polymerase has. Sometimes I say the DNA polymerase is like undergrads, it's got its limitations, and then the RNA polymerase is more like a grad student. So now, zooming in a little bit on the polymerase and what you see happening, there are a few different polymerases you're going to see named throughout this lecture. The bacterial DNA polymerase 3, that's the one that's actually building new strands of the DNA for the bacteria. When you look at this figure, you see some of the key players that we talked about already. You see the gyrase uncoiling the circles of DNA. You see the helicase unzipping the double strands. You see single-stranded binding protein, which is SSB, coating the single strands to prevent them from religating. And then you see the DNA polymerase, and you'll see that it's made of several subunits. It has a core enzyme. It has a sliding clamp to allow it to hook around those single strands, and it has a clamp loader or clamp loading complex. And when you look at it, you notice that one of the strands is looping out like this. That's because the lagging strand is in the opposite direction of what the polymerase needs, because the polymerase must build 5' to 3', and it wants to build the leading and lagging at the same time, but the lagging is in the wrong direction. So it loops the DNA of the lagging strand around like that so that it can build in the proper orientation. And that's why you see, Okazaki fragments or the lagging strand built in little segments. Now, what's valuable about this polymerase 3 in bacteria is it also has what's called kinetic proofreading and mismatch repair. So basically, the proofreading senses a mismatch because of the distortion of the shape of the DNA molecule. So basically, if you have the wrong base placed in a certain location, the polymerase will say, hey, this is not the right, width of the double-stranded DNA. There's a problem here, and it has an exonuclease subunit, which allows it to cut out the wrong bases, cut out the mismatch, and then get back to work repairing it and putting the proper bases. Okay, so you'll notice that since it is made up of multiple subunits, you have a listing here of some of the ones that we just mentioned with their official little subunit name. Okay, so now once you've had replication occur, there's a big problem at the end because if you remember, primase was an RNA polymerase. And so those primers that you have are actually RNA bases. You don't want RNA bases in your DNA because remember, RNA is going to have uracil or the U, whereas DNA is going to have T, thymine. And so you'll completely mess up the code if you have a RNA bases there. So that's the problem. You have RNA primers, RNA bases in your DNA at this point, or in the bacteria's DNA that we were just looking at bacterial replication. So now there are three enzymes used to correct this in order. The first one is RNase H, which is not shown in this figure, but basically RNase H is going to degrade or break down a major portion of that RNA primer. And then pol 1 comes along and it's a polymerase, it's a DNA polymerase. So it's going to start replacing the missing or deleted RNA bases with DNA bases because it's DNA polymerase. And then at the end, you have DNA ligase that's going to seal up the backbone, okay? Fix any of the nicks in that DNA now. And so kind of gluing everything together, okay? So as a quick recap of all of the process, The first thing that comes along in bacterial DNA replication is the gyrase to uncoil that big ball of condensed DNA. Then you have the helicase to unzip those genes, lots of memes about that, and make them the double stranded DNA into the opened up single stranded templates. You then have SSB, single strand binding protein, to coat those single strands and prevent them from religating together, sticking back together. You then have the polymerase come along, but because of the problem of that DNA polymerase not being able to start from scratch, start strands, you first need RNA polymerase. So you need primase to make the RNA primers, and then DNA polymerase to make the rest of the DNA strand. At the very end, you then need the RNase H to break down or degrade the major portion of those RNA primers. Then DNA polymerase substitutes them with DNA bases, and ligase glues up the backbone, sticks everything together, cleans up the mess type of situation. So now all of that was the general overview of DNA replication that you kind of see in genetics and in general bio and everything. I want to give you a little more of the biochemistry side of the process, looking a little bit more at the biochemical mechanism of DNA polymerase. And so when we look at DNA polymerase, the reaction that it has occurring is going to add a deoxynucleotide to the 3' end of a growing DNA chain. Okay, and so over here you see 5' end is up here, 3' end is over here, and you're going to have the direction of synthesis going in this order where you have the addition to the 3' end. And what you will notice is that when you have this occurring, because DNA polymerase is an enzyme, you're going to need a cofactor to help it, be activated and function. And so in the case of DNA polymerase, what you end up needing is magnesium as the cofactor. So magnesium ions will basically stabilize the negative charges on the deoxynucleotide and assist in the linking of that, 3' OH with the base joining over here and having that linkage that you see occurring at the 3' end. Okay? And so when you see here, you see the magnesium cofactors here helping in terms of that process to get the base added to this 3' OH here. And this is what was the 3' OH now has the next base linked to it. Okay? And so when we look at this process, I want to point out that very critical in terms of DNA polymerase activity is the active site. And so the active site, the shape of the DNA polymerase active site is going to facilitate the formation of the correct Watson-Crick base pairs. And so you see that over here. So the correct base pairing is GC and AT, right? And so when you look at the active site, it's basically the perfect shape and organization to have a purine and a pyrimidine linked together so that you ultimately have, you can think of it as a 3-ring structure within there. And so the active site, if instead you get a mismatch, incorrect pairs, they're not going to fit well into the active site pocket, as you can see here. If instead, for instance, you have two, single bases and you have the, not the right amount of rings, not the right, side groups on them, you're not going to have a good fit into that active site and they end up largely excluded. Okay. And so when you have that happening, the active site is going to prevent you from having the formation of the wrong pairing. Now, as I mentioned earlier, when we looked at the DNA polymerases, we focused on bacteria polymerase number 3. But I mentioned that you have a lot of different polymerases that you're going to see the names of. So these are the three major ones in E. coli and the major function. So I want you to circle this top portion here, circle star highlight what each of them does, what they're known for. And then these are the eukaryotic ones. And I want you to also circle star highlight the name of the polymerase and its function. So with eukaryotes, you see that the one we emphasize so far is polymerase 3, and pol 3 is the main polymerizing enzyme, meaning that's the one that's going to be building the new strands of DNA. Okay. You then notice that pol 1 has proofreading, repair, and primer removal. So we saw it in the first slide of the bacterial DNA replication when we had the full look at the polymerase, but we also saw it again at the end when we were removing the primers, and it had to pole one had to replace the removed primers with the proper DNA bases. And then pole two is involved in repair. Okay. When we look at the eukaryotic ones, pole delta, which is this first one here, that's going to be responsible for synthesis of the lagging strand, whereas pole epsilon will be responsible for the leading strand synthesis. Okay, so in bacteria, pole 3 did both leading and lagging strand. In eukaryotes, you have delta dealing, synthesizing lagging, epsilon synthesizing leading, and then alpha, alpha being the first in the alphabet, that's the initial synthesis from the primer. That's the alpha you can think of as initiation primase kind of function for it. Okay, so make sure that you know each of the eukaryotic ones and each of the bacterial DNA polymerases, what the name is and what the function is. And make sure to know that if, for instance, I mentioned pol delta, you should know that that's eukaryotic and not bacteria, not, for instance, in E. coli. And on your chalkboard notes summarizing those three from each organism, I also added a nice little figure that I think helps remind you of some of the key players in the process. Now, I want to mention a very important other enzyme that some organisms have, and that's reverse transcriptase. And so, if you think about what transcription is, that's DNA to RNA. And so, if it's reverse transcription or reverse transcriptase, that means you're going in the opposite direction. So, the function of this enzyme is to copy RNA to DNA. And you see that used, for instance, where? Well, in HIV replication. And so this ends up being a big problem. Any viruses, for instance, that use reverse transcriptase and they make a DNA copy of their RNA genome, that becomes a big problem for us because reverse transcriptase is highly prone to error and there's no proofreading function. I basically say that this enzyme is the equivalent of my undergrads, makes a ton of errors and does not know how to proofread. And so when you look at that, that means that, well, if it's making a lot of errors while it's making a DNA code, what do we call those errors? Mutations. And that's why it becomes so problematic to try and create cures or vaccines or preventions for some of the viruses because they create so many mutations when they're changing or making a copy, a DNA copy of their RNA, that in our bodies, by the time we would give that drug or that vaccine, the virus has made so many mutations that it's got a new code and, that drug or whatever would not work anymore for it. And so that's something that I really want you to remember with reverse transcriptase. So even though it has a similar structure to DNA pol 1, this guy makes a lot of mistakes. So now, when we talk about DNA replication, when you think about, well, when is a cell going to replicate its DNA? Well, right before it wants to divide, right? Because it's going to need to double its DNA so that it can divide and so that each new cell has the proper amount of DNA. That means that the replication process must be synchronized with cell division. You need a specific start and a specific end. Now, we already mentioned that in order for DNA replication to occur, you need an origin of replication, and that is the start site. Then you have what's called DNA A, C, and B. What DNA A does is that it binds to the origin of replication, so OREC, and it opens the DNA up. Okay, so it's going to basically have a helicase function or a function to start opening that DNA, or almost more like a gyrase function as well to kind of expand it out. And so once you have that occurring, you then have DNA C proteins that I'm going to find it over here. Okay, so over here you have DNA C, which helps DNA B bind to this region where you're starting to have DNA replication start. And DNA B is the real helicase proteins that need to unzip the DNA. And basically you have these subunits here helping to create the replication fork. And when that happens, you're now displacing DNA A so that DNA B can take over, with its helicase activity. Then we go through the steps that we mentioned previously where you have primase, and the primase is going to make the RNA primers. You have the official DNA gyrase and SSB that we mentioned before, so kind of uncoiling the big condensed DNA. You have SSB, which is preventing religation of single strands. So you basically have a whole bunch of different enzymes and different proteins working together to have DNA replication occur. And I want to point out, in this process, you see that DNA A is initiation. It's involved in initiation. And again, you can think of kind of like what we said with alpha. Think of A as being the first letter of the alphabet. So that one will be for initiation. It basically helps loosen DNA in 13 base segments and kind of thus allows DNA B to be recruited to work as a helicase and then bind thanks to DNA C, which you can think of as a helicase loader protein. So the DNA C is simply there to load the helicase onto the DNA. Now, with anything in biology, not only does it have to be highly synchronized, but it has to be highly controlled. You don't want, replication just occurring all the time. You just want it before cells divide. And so basically, when you think about this process, we said that DNA A is the initiator of the process. Now, DNA A binds better to fully methylated DNA. What fully methylated DNA means is that both of the double strands are methylated. They have a CH3 group on a particular sequence at the origin of replication. So it's the GATC sequence of the OREC. Again, OREC is the origin of replication. So DNA binds better when you have this spot on the origin of replication fully methylated, okay? Rather than at the end of replication, what you notice is the original parent strands have the methylation, but the newly synthesized DNA does not. It has not been methylated yet. And so we call this hemimethylated, so half methylated DNA. DNA does not like to bind to that. It doesn't see that as a good recognition site, okay? So what happens is you have regulation because the something called the SEC-A protein, SEQA protein, will bind this hemimethylated origin of replication right after replication, and it will sequester this hemimethylated DNA to the cell membrane. Okay? Now, that sequestering, pulling this to the cell membrane area, prevents the binding of the initiator DNA, which does not like this kind of hemimethylation. Now, as you have the growth phase of the cell, okay, that cell membrane is going to be expanding, expanding, and moving. Well, now at a certain point, you get the release of that origin of replication from its sequestering. Okay, so during the growth phase, right before the cell splits, you get that release. Okay, it is freed, and now this hemimethylated DNA that has been released from being sequestered, now it can be targeted by those damn methylases. And I love saying that because it's actually DAM, that's their real name, and it can be targeted by those methylases. So now that double stranded DNA is fully methylated. And what likes fully methylated DNA? DNA, the initiator of DNA replication. Okay, that binds, to that fully methylated DNA rather than the hemimethylated one. Okay, and that's to prevent that whole process here that we just talked about, and that sequestering by Sec-A is to prevent the immediate refiring. Okay, so when I say how is this prevented, it's that Sec-A binds the hemimethylated new double-stranded DNA, sequesters it at the cell membrane until ultimately you get that, growth phase and cell division, and then you can now get that methylation, and DNA can now initiate DNA replication next time. So now, as I was just saying about everything having to be synchronized and have control in science, which is great because, us scientists, we want control too. But anyway, you need to have processes highly regulated. You don't want things to start and not be able to stop. You don't want things uncontrollably starting or uncontrollably ending. So to everything, you need a proper end, which sounds morbid or sad. But for DNA replication with prokaryotes, termination is guided by what's called the TER-TUS complex. Okay, so if you think about it, replication is ended by blocking DNA B, which is that helicase that we mentioned a few slides ago, via the TER-TUS complex, whereas refiring was prevented in the previous slides by blocking DNA. Okay, so replication is terminated by blocking DNA B via TER-TUS sites, and I'll have the spelling of that on the next chalkboard slide. Whereas previously we mentioned refiring is prevented by blocking DNA. So I want to just show you how the TER-TUS sites end up blocking DNA replication and blocking that DNA B. Basically, what you'll notice is the TER sites are on the DNA. They are sequences that bind TUS proteins. Now, when you look at this structure here, notice origin of replication is over here. You have bidirectional replication, so you have replication going this direction, and at the same time, replication going this direction, and the termination is going to stop here. Now, the way TER-TUS ends up blocking or allowing for termination is that these proteins, these TUS proteins, when you think of this region basically, okay, it's one directional. Picture that at these TER-TUS sites, this side is an entrance, this is an exit. Entrance, exit, entrance, exit. So over here, picture you have an exit at this end here. Well, when this direction is going here, entrance, exit, entrance, exit, entrance, exit, it hits an exit. So DNA B can't continue on in this way. Okay, basically what happens is you can picture the two the two replication forks, they end up colliding over here. Okay, so you have termination when those forks collide. Now, when you have this occurring and you have termination, what you see here is you have, because it was bidirectional and it's ending over here, and circular DNA because it's prokaryotes, you have this tangled connection of the daughter chromosomes. And so, what you need to happen at the end of this termination is you need to decatenate these daughter chromosomes. And that's a fancy way of saying detangle or separate the daughter chromosomes, okay? Because otherwise, you're going to have crossing over between the interlocked daughters and you're going to have two DNA strands stuck together. So you need to decatenate or separate, detangle and separate the daughter chromosome so that you have two distinct, separate rings of DNA. Okay, that was all prokaryotic or bacterial. So I just want to show you briefly that you also have eukaryotic DNA replication. It looks very, very similar. The main difference here is that you have the DNA is now linear and longer than bacterial DNA. You're going to have multiple origins of replication. You're not going to have things like gyrase. Notice there's not going to be any gyrase up there, but ultimately, the process looks very similar, just with different names. So, for instance, over here, you have MCM, which in the bacteria, this is separating the strands, so helicase activity, like DNA B, which was the helicase in bacteria. You still see, these green circles, which were previously called SSB. In eukaryotes, we call them RPA for replication protein A. Okay, so put a little star next to MCM and write that that's the equivalent, that's the eukaryotic equivalent of the bacterial DNA B helicase. Put a little star next to RPA and write that that's the eukaryotic equivalent of the bacterial SSB. Okay, you notice now that the sliding clamp is called here, the sliding clamp loader is now RFC for replication factor C. You also have a lot of other um, letters that are different, but the overall process is very similar. Notice that you don't have a dual function DNA polymerase, DNA polymerase 1, the way that you had in the bacteria. Remember, we mentioned in eukaryotes, you have DNA polymerase delta, you have epsilon on the other side taking care of the other strand. Ultimately, in eukaryotes, you'll have the primers removed by exonucleases and gaps are filled by the polymerase delta, which you see over here. So this is showing what we saw at the very end of bacterial DNA. This is how it's going to happen in eukaryotes. And then at the end, just like bacteria, you end up having a ligase seal up all of the final little gaps. Now, I want to point out that since we mentioned that eukaryotic DNA is linear, then, that big difference of eukaryotic DNA being linear instead of circular, the way the bacteria had, now you need telomeres. And basically, that's long sequences at the end of the DNA that are non coding. And the reason that is when you have linear DNA, getting replicated. Each round of replication, when you have the removal of primers in linear DNA, the RNA primers, and you replace them with DNA, at the very end of the linear DNA, there's no bases on the other side of it, the way that a circle would have. And so the DNA polymerase can't replace that very end primer, right? Because we said DNA polymerase needs, it can't start from scratch. It needs something else to link to. So with every round of eukaryotic DNA replication, you lose some of the end of the DNA. But it's okay when you have the telomere there because you're just losing bases that are non coding. They're not going to code for anything, you're not going to be missing anything. But eventually, you end up hitting genes that code for something. And so that's kind of why you get aging and why telomeres are looked into, the shortening of these telomeres are looked into as a major factor in aging in, for instance, humans. Now, some cells do have what's called telomerase, which you see in this figure, can replace the missing bases, but that in humans is only, limited to undifferentiated embryonic stem cells, male germ cells, which means sperm, and activated lymphocytes. So the rest of our cells do not have telomerase to replace those ends, so we get shortening of the telomeres and aging. I now want to switch over to DNA damage and look at mutations. So, when we talk about mutation, basically that's a permanent change in a DNA sequence. It can end up possibly affecting gene function depending on which DNA sequence it is, and it can alter protein coding sequences, which can be very problematic. Now, if it occurs in somatic areas, somatic cells, those are not genetically inherited. Okay, so if a mutation occurs in a germ cell, meaning your gametes, your sperm, your eggs, then the mutation gets passed on to your offspring. If it's a somatic mutation, such as let's say your father had skin cancer, in a somatic cell, you are not going to be born with that mutation, with the mutated skin cells. Okay, now ultimately, when we look at this slide, the 10 major types of DNA damage are summarized in this figure here. So, abnormal base pairs, chemical addicts, base pair mismatches, double strand breaks, single strand breaks, abasic, site, for instance, missing a base, thymine dimers, so two bases linked together, DNA cross-links, nucleotide insertions, and nucleotide deletions. And we're going to go through some of those in a moment. And ultimately, if you have certain mutations, an accumulation of mutations, you can end up with cancer progression. And, a lot of times mutations end up in genes that are regulators of cell division, and you end up with uncontrolled cell growth and replication, which ultimately is cancer. You get the formation of, of tumors. Now, we're not going to go into crazy detail with the Ames test. I just want you to know that the purpose of an Ames test is to determine if a suspected mutagen, meaning if you think that a compound, a chemical, is a mutagen, that it creates mutations in cells, then you can do the Ames test. And basically, what it is that they mix that test compound with um, a special type of mutated bacteria and rat liver extract. And the mutation that the bacteria currently has is where it's his gene. It's mutated, so it cannot produce the essential histidine. And so then, if you combine it with the test compound, and the compound reverses that mutation, meaning that it mutates the mutation, you then end up with bacteria that can produce histidine. And they show that based on growing the bacteria on plates that are lacking. They're called minimal media that's lacking histidine. So the original bacteria would die on these plates. You would not get bacterial growth, you wouldn't see spots on it. Whereas if the test compound is a mutagen, you see a whole lot of growth because that compound is mutating the bacteria so it can grow now. So now I want to go into some specific examples of DNA mutations. First, I want to point out the possibility of frame shift issues. So if a DNA mutation basically occurs where you have an addition or a deletion of bases that are not a multiple of three, because as you know, DNA is read as codons, okay? Sets of three bases that are then read as a codon of RNA that then gets translated into a particular amino acid. So it's read three letters at a time. If your mutation that occurs is not a multiple of three, meaning let's say only one base got deleted or inserted, or two bases, or four or five, okay? Because multiples of three would be 3, 6, 9, 12, for instance. If the mutation is not a multiple of three, well, now everything after that mutation is going to be read by the cell wrong. The cell doesn't have eyes to say, oh, we're missing one base here. Let me, make note that this codon is not complete. No, it's just going to start reading the next letters. It reads three letters at a time. Now, everything past that point will be wrong. Okay, so frame shifts can be really, really bad. Now, when we talk about mutations, some of the examples I want you to know are missense, nonsense, and silent mutations. Missense mutation, that's a single point mutation, a single base mutation in which one letter has been changed. Okay, so you're not deleting or inserting a letter, it's a single base mutation where one letter is now changed to another letter. In missense mutations, which are the middle examples, you now end up with a different amino acid than you're supposed to have at that location. So here you were supposed to have a lysine, missense occurs, and boom, now you have an arginine. Or a missense occurs, and now you have threonine. The reason it shows two different possible missense mutations is they could either be radical, which is non-conservative. So non-conservative means the same thing as radical. When you think of radical, that means really different. If someone or something is radical, really different. So that means that the amino acid that is now in that location is very different from the one that was supposed to be there. So notice the one that was supposed to be there was supposed to have a positive charge. If the mutated one now has either a negative charge or no charge at all, that's very different from the chemical and physical properties that you're supposed to have there. So that is a radical or non-conservative missense mutation. If, however, the new amino acid has the same chemical or physical properties, so it's supposed to have a positive charge, it still has one, a positive charge, that's conservative, didn't really change much. Conservatives in terms of people, they don't want change, they want traditional. So you can use that to help you remember the difference between radical versus conservative. Now, notice this is a big deal in proteins because, for instance, if you have a non-conservative radical missense mutation, you were supposed to have a positive charge there. You don't now. That greatly affects the interactions and the folding of that protein, okay? Because positive will want to interact with negatives, and now you don't have that in that location. You can also have nonsense mutations. And so I always tell my students when you see nonsense, think stop the nonsense. Nonsense mutation means instead of an amino acid, at that mutation, it caused a stop codon. So you have premature termination of that protein. Okay, you won't have anything after that. You now have a stop codon. The last option is a silent mutation over here, where yes, one base letter was changed, but you don't have anything different. You don't have a change in the amino acid that was supposed to be there. So it's a silent mutation, and that's possible because, as you know, the genetic code is degenerate, meaning that there's a lot more codons possible than there are the 20 amino acids. Okay, now that we have taken a look at some of the possible issues that you could have in DNA, the mutations, I want to go through some of the repair mechanisms. Now, I'm not going to make you memorize the crazy details about them, but I want you to know the key players and kind of what's different in each of them. So, over here, when we have the prokaryotic mismatch repair mechanism, so mismatch repair, the proteins involved are going to be what I call the MUT-S, MUT-L, MUT-H uh, complex. So you'll see MUTS, you'll see MUTL, and they form the MUTS, MUTL, MUTH complex for mediating that mismatch repair. Basically, the MUT-S binds to the mismatch mutation. So MUT-S is going to be the first one, and that leads to the subsequent steps, beginning with the ATP depending binding of, where is it, MUT L. So now you have MUT L over here as well. So you see that here. Um, and with that, you get the rearrangement of the DNA to basically accommodate the formation of that MUT-S, MUT-L, MUT-H complex that will be over here responsible for endonuclease and exonuclease activity to remove the problematic DNA section. And you see that single-stranded DNA binding proteins will also be involved to help prevent, any sticking of foreign DNA that doesn't belong there, to give time for DNA pol 3 and DNA ligase to repair the gap of that missing DNA that it basically, the mismatch repair, cut out from here. Now, over here, you're going to see two different mechanisms. We have base excision repair and we have nucleotide excision repair. And base excision repair is basically mediated by what's called either short patch or long patch repair. So there are two different possible pathways here. The short patch pathway, you'll see, basically removes only one nucleotide, whereas the long patch will remove up to 10 nucleotides. And basically, what you have here gets initiated by spontaneous cytosine deamination. So that'll be the initiation signal to have either one of these pathways initiated. And when we look at this then compared to nucleotide excision is going to be used for large lesions that are basically distorting the helical nature of DNA, which can be problematic. Instead of being initiated by the cytosine deamination, this one, nucleotide excision repair, is initiated by the recognition of the lesions by what's called UVR AB. Okay, so UVRA and UVRB, you can think of it as UVRAB is going to recognize and initiate, usually trying to fix something like a thymine dimer over here. And with this one, then the nicks in DNA are created by UVRB, UVRC, and then finally you have UVRD down here, which is a helicase, and that will remove the polynucleotide that contains the lesion. And so you see that you have this basically segment of DNA getting removed here. And then, as we saw with other situations, you have pol 1 and ligase fixing the gap. Now, I just want to point out the big difference between these two things is that the base excision repair pathway, that's correcting only the damaged bases, whereas the nucleotide excision repair, that's correcting bulky DNA adducts, as we call them, and it's going to remove a whole segment, a short single stranded DNA segment, along with the actual lesion or problematic section. Now, I also want to mention that DNA photolyase exists, and that's another DNA repair mechanism. I'm not going to go into the details of this one. Instead, I just want you to know the purpose. When you see photolyase, you know, or DNA repair by photolyase, just know that this is to repair sun-induced pyrimidine dimers. So, for instance, sun-induced thymine dimers. The last forms of DNA repair that I want to mention are single strand break repairs and double strand break repairs. And so basically, when you look at the single strand DNA break repairs in eukaryotes, it's very similar to the base excision repair and nucleotide excision repair mechanisms, but basically will have different names for the proteins, okay? But the gap remaining, what you see here is you end up having with a short repair gap filling path, a long repair gap filling path, and you see some of the enzymes that we mentioned previously, things like pol delta, pol epsilon. Over here you have pol beta, and again, you constantly see that ligase has to be involved in any repair mechanism because you need to seal up the final backbone gaps that you have. Now, double strand DNA break, what I want you to remember for that one is it's going to be repaired by what's called homologous recombination or non-homologous end joining. And we're going to go through in a moment, what homologous recombination ultimately entails. But here you see in this pathway, you have the DNA repair enzymes RAD51 over here. You have BRCA1 and BRCA2. Okay, all of these are DNA repair proteins. They'll initiate the repair, they'll form a complex, and then ultimately you have them forming a homologous duplex, and you end up having DNA synthesis, which will use the homologous template strands to replace what's missing, and you end up with a repaired strand. And ultimately, DNA polymerase is going to replace any, missing strand that you have during the process. Okay, but again, you don't need to know the crazy details. It's just to kind of show you some more possible repair mechanisms. The last thing I want to mention in this lecture is DNA recombination. So we just mentioned that, for instance, homologous recombination is involved in double strand DNA breaks. So I want you to kind of see DNA recombination. DNA recombination events naturally occur during meiosis, and they're very, very important when creating gametes during meiosis because it introduces genetic diversity. And so you see, let me get the laser pointer, you see here that during meiosis, you can basically have crossover of segments of chromosomes that are homologous to each other. You get the exchange of those segments, the exchange of that DNA. And now, when you have the segregation of those chromatids, and ultimately you have the production of gametes, you could end up with new genotypes, new, diversity. And that's very valuable to us. Okay, but you'll notice that DNA has to have a double strand break in order for this to occur, and you see that this is focusing on homologous regions. Now, when you have recombination, you have something called a Holliday Junction occurring. In genetics, you get to see videos or more detailed kind of description of all of this. But for our purposes in biochem, I just want to show you briefly that the Holliday Junction is basically what they call a quadruplex DNA. So there are four different DNA strands coming together here, and you end up having branch migration possible where the Holliday junction can basically be moved along the chromosomes by helicase activity. And then ultimately, this Holliday junction is resolved by what's called resolvase. Okay, resolvase is going to, um, you know, make sure that you get rid of the Holliday junction and you end up seeing that your ultimate final products, I guess you would call them, can have crossover where the four different strands of DNA that you had over here, now you have, mixtures of them in the final product. Okay. And so again, we don't go into the details that we would in genetics for this, but just notice that it's giving, when you see recombination, you get a new mixture, you can kind of call it, of the DNA. And so that gives diversity. And I want to circle a star, highlight one of the big recombination proteins over here, SPO11, since my name is Esposito and my nickname is Espo or SPO. I always like SPO11. So circle star SPO11 to let yourself remember that whenever you see that, that's involved in recombination. Okay, that's going to be important for initiating the recombination. And whenever you see Holliday Junction, spelled just like Doc Holliday of Tombstone, whenever you see Holliday Junctions, that's also a characteristic that I want you to associate with recombination. And so here is a review question from today's lecture. Please, again, the moment that you get to this question, I don't care if it's 3 in the morning, 4 in the morning, whatever. I don't care if it's a week after you were supposed to watch this video, but now you're deciding to study that there's a test the next day or something. Just send me this slide as soon as you get to this point. Take a picture of the slide and send me your answer in the Remind app. Or if you're someone not in my class and you want to test yourself and see if you're correct, you're always welcome to send me an email or comment to kind of go through any of the questions that you had during this video. That is it for today. Thank you. And if you have any questions, never hesitate to contact me. Always, please, if you can, send me a picture of the slide to help me best explain when you are asking a question. Thank you and have a great day.